All right, welcome everyone. Thank you for coming to our 2021 Vancouver Bioinformatics User Group 3-minute lightning talks. We're really excited. We've got 14 speakers here for you today to talk bioinformatics. Give you a little overview of what the schedule looks like. I'm going to give a little introduction and talk a bit more in detail about the overview. Then we'll have about 50 minutes for the talks themselves. We're going to then have a viewer's choice poll, the question and answer period, and lastly, we will announce the winners as well as the audience prize draw. So how this presentation is going to work is that every speaker has a limit of 3 minutes to present, and then they will be cut off at the end of those 3 minutes. They're going to have one static presentation slide, so don't be judging them for flashy animations or videos because we didn't let them put anything like that in there. And then we have 2 judges who will be judging on clarity, scientific soundness, delivery, the use of visual aid, and overall impression. And you can use the same criteria to evaluate as well if you would like. Thank you to our volunteer judging panel. We really appreciate having you both here. We have Dr. William Zhao, Associate Professor at Simon Fraser University in the Faculty of Health Sciences, and also the PI for the Center for Infectious Disease Genomics and One Health, as well as Dr. Wyeth Wasserman, Professor of the Department of Medical Genetics at UBC, the University of British Columbia, and also affiliated with BC Children's Hospital. We couldn't be doing this without you, and we really appreciate having you here. As for the prizes, we have gift certificate and monetary prizes that will be going out. For third place will be $45, second place will be $90, and first place will be $150. And these will be for gift certificates for your choice of location, as long as it's in Canadian. Then there will be the Viewer's Choice Award, and this is the award that you will be, the audience, will be engaging with. So at the end, once everyone's spoken, we'll provide you a link to a poll. You can pick who your favorite was, and that's who this is going to go to. Even if they won another prize, this is going to be a sincere, like this is your choice that we're putting out here. And then anyone who enters into the Viewer's Choice poll will then be entered into the audience prize draw. And we have 2 different $30 gift certificates that will go to one of you. So VanBUG is sponsored by the Graduate Studies Program in Bioinformatics through SFU and UBC, the Genome BC, Ecoscope, the Canadian Bioinformatics Workshops at bioinformatics.ca, AbCellera, Langara College, Stem Cell Technologies, as well as other bioinformatic user group affiliations. We have the International Society for Computational Biology, the Montreal Bioinformatics User Group, and the Toronto Bioinformatics User Group. Here's a little rundown of what things are going to look like. I'm going to introduce the speaker and the title of their presentation, and then I will say speaker or speaker's name. You may begin. That is when the timer starts. They will have exactly 3 minutes before they will then be cut off if they continue to talk. And then I'm going to move on and go to the next introduction slide and introduce them and rinse and repeat. So please hold your questions until after all speakers have presented. But especially if you're asking a question in the chat, but also verbally, please make a note of who your speaker's name is or number so that we actually know who it's being directed to and a little less confusion. This is our roster for tonight. You can see the numbers here. You don't have to have that memorized, but thank you all of you speakers for coming and doing this. We're so grateful to have you here. It wouldn't be happening without you. And now I guess I'll just get rolling. With our first speaker, we have Armita Safa from the Bioinformatics program at UBC under the supervision of Dr. Faraz Hash. She will be talking about RinsLR, the discovery of mid-range novel sequence insertions using long-read sequencing. Speaker, you may begin.
Hello, everyone. Genomic variations are differences in the DNA of individual members of a species, and they can provide insights into evolutionary processes, susceptibility to diseases, genomic diversity, and ultimately help us better understand the human genome. In this project, we focus on one class of variants called novel sequence insertions, which are inserted sequences that are not found in the reference genome. This class of variants are harder to detect because there's a missing sequence that needs to be reconstructed and thus has the classic issues of assembly such as repeat region resolution, and using long reads can help us overcome these problems. Our goal here is to develop a tool that can detect novel sequence insertions with good accuracy. RinsLR, our tool, first analyzes the alignment of short reads to the reference genome to identify potential breakpoint locations through signals such as soft clippings. And if a certain location has many reads that are in favor of it, we will mark it as a potential breakpoint. In the next step, we create a fingerprint index of the long reads, which is an approximate summary consisting of minimizers on the reads and the locations in which these minimizers are found. Therefore, we can rapidly query it and get approximate answers. Then we extract the genomic anchors for each side of the breakpoint and use it as the query to the index. This will provide us with a set of long reads that are potentially mapping to this region. These reads are either picked from the left side or the right side or from both sides, which we call bimodal reads. We also do fast approximate local chaining from the long reads to the genomic anchors, which are the bubbles on the reads, to ensure that they actually provide a good mapping, and it will also help us estimate the insertion lengths through the distance between the chains on both sides. And will also help us identify reference allele supporting reads that we can discard because they will not help us construct the insertion. Then based on the locations of the chains, we identify an interval in each read that is mapping to the genomic anchor, as well as the part that covers the potential insertion. We trim the reads accordingly. Now, having our trimmed and classified reads, we create a multiple sequence alignment of the reads, and then based on the MSA and profiling information from it, we build a high quality consensus. Now, for identifying the actual insertion, we do a semi-global alignment from the constructed sequence to the genomic region around the breakpoint. And therefore, we can discover the exact breakpoint location along with the insertion content. And then we can move on to downstream analysis, which would include genotyping of the insertion. Thank you very much.
Wonderful, thank you so much. Okay, our next speaker is Asfar Lathif Salaudeen, under the supervision of Dr. Karl Huebert. I apologize if I pronounced that incorrectly, under the School of Biomedical Engineering at UBC. He will be talking about deciphering gene regulatory grammar in cancers. Speaker, you may begin.
Hello, everyone. Mutations in the genome are one of the major driving forces of many diseases, including cancer. Mutations that occur in the coding regions of genome are relatively well characterized because of their effect on the gene products and how it directly affects the phenotype. But unlike these mutations that occur in the non-coding part of the genome, like promoters and enhancers, are not very well characterized. This is mainly due to the lack of data to associate these mutations with their corresponding phenotype. The goal of my project is to build machine learning models that can understand and predict how different sequence variation in the regulatory regions can affect and cause cancers. To build these models, we would require a vast and diverse training data of sequence and their corresponding expression level. This is a huge limitation now because of the few observed and characterized non-coding mutations. To overcome this issue, my project will be involved harnessing the power of synthetic biology to randomly mutate the regulatory regions of interest using CRISPR-Cas9 based tools like base editors, which can create random mutations in the genome. And from these mutations, the independent set of mutations that are created in millions of cells can affect the gene expression in these cells by either upregulating or downregulating it. And once we have these various gene expression level, we'll be able to sort these cells into multiple different bins. And once we have that, we will be able to sequence the particular target region and match that sequence to the corresponding expression level. This will give us a sequence to expression data that can be used to train machine learning models on. Using this training data, I'll be building an interpretable biochemical model that takes into account the known transcription factor biochemistry and will be able to understand how the variation can change the dynamics of transcription factors binding in that area, and thus giving us an overview of what exactly is happening in that gene regulatory network. Besides that, I'll also be building a neural network using this data, which helps us to estimate how a particular mutation in this sequence can change gene expression level. This will be very useful for us in predicting how new oncogenic mutations arise, and is also very helpful in understanding clonal evolution and how tumors can progress in a microenvironment. To summarize, I'll be building machine learning models using data obtained from randomly mutating regulatory regions and measuring their expression level, and using that to predict how mutations can cause and affect cancer phenotypes. Thank you.
Wonderful. Thank you so much. All right, our next speaker is David Huebert, under the supervision of Dr. Mani Laranjani at the Simon Fraser University Molecular Biology and Biochemistry program. And the talk will be Mistakes of the Past and the Evolutionary Immunology of ApoBec 3G. Speaker, you may begin.
Mistakes are integral to life. Mistakes created both the benefits of things like microwaves, the devastation of Chernobyl, but within us and across all primates are ApoBec proteins that can cause mistakes. Now, ApoBec 3G stops viruses going from one species to another by mutating the viral DNA to the point of its destruction. However, some viruses, like HIV and SIV, which is in primates, have found a way around this. They created a protein called VIF, which is able to destroy the ApoBecs, first by binding them and leading them to degradation machinery. And they increase the chances that over time beneficial mutations will happen to the virus that allow it to escape our immune system and escape any drugs that we try to make against it in the future. To understand this evolution, we truly need to understand the true structure of these proteins. Unfortunately, through chemical methods, we've never been able to expand it to such a size to be able to understand the evolution of all primate and viral structures. I utilize computational methods like ITASR to create libraries of hundreds of these proteins to see them in 3 dimensions across evolution. I noted the immense differences in their surface structures and how their cores are maintained across all primates. To give you an idea of what this is, the difference between all the primates is equivalent, in this one protein, is equivalent to the difference between you and a fish. Massive variation. This made us wonder what the surface charges are. This allows us to understand how proteins interact. And while there are a lot of local changes and variations in charge due to evolutionary pressure, overall, these proteins are highly well maintained to allow them to get around our all of the different environments that they survive within. We also used haddock to map the interactions between these proteins, docking them with one another. And we found that the same 3D locations of binding are maintained across the evolutionary pressure, but through drastically different chemical means. Finally, we've created hundreds of these evolutionary structures. We've noted the massive variations in between them. We've noted differential surface charges. We've found consistent evolutionary interactions. By studying the immunological coevolution of ApoBec 3G and VIF, we're able to track where mistakes have occurred in the past. And by that, we may predict future mistakes or even direct mistakes for ourselves. Or maybe we'll just be mistaken. Thank you very much.
Thank you. Okay, and our next speaker is Denitsa Vasileva, under the supervision of Dr. Dennis Daly. She is a member of the bioinformatics program at UBC and will be talking about epigenetic age prediction using targeted methylation sequencing data. Speaker, you may begin.
Aging is a complex process which proceeds differently in different individuals. Some people experience cognitive decline and physiological frailty much more quickly than others. The current metric which we use to measure aging, chronological age, or the number of years that have passed since birth, does not accurately account for this heterogeneity. This has led to an interest in the development of tools which can more accurately measure the aging process. These are known as aging biomarkers. The epigenetic clock has been repeatedly independently validated as a promising biomarker of aging. Epigenetic means above genetics and refers to heritable changes in gene expression which are not caused by changes in the DNA sequence itself. The epigenetic clock leverages the changes in patterns of an epigenetic modification known as DNA methylation over the lifetime in order to accurately predict epigenetic age. DNA methylation involves the placement of a methyl group, a CH3 group, on the 5th carbon of a cytosine base by an enzyme called DNA methyltransferase. In humans, this usually occurs at what are known as CpG dinucleotides. So this is when you have a cytosine and then a phosphate linkage to a guanine base. In general, epigenetic clocks are developed using penalized linear regression and consist of a list of age-informative CpG sites together with a mathematical algorithm. The difference between the predicted epigenetic age and the reported chronological age has been shown to have good predictive power, especially when predicting outcomes such as mortality or the risk of developing various diseases from asthma to cancer. Many epigenetic clocks have so far been developed, but the Huebert age prediction algorithm is the most widely used. It was developed using elastic net regression on adult methylation data from Illumina arrays. However, its accuracy in pediatric samples and when using methylation sequencing data has not been extensively studied. In my thesis, I used a unique data set of methylation sequencing data from 932 samples from 3 Canadian studies: the Canadian Asthma Primary Prevention Study, or CAPS, the Saguenay-Lac-Saint-Jean study, and the Canadian Peanut Allergy Registry, or CAMPAC. CAPS is a longitudinal birth cohort, which follows children who had high risk of developing asthma from birth to year 15, with methylation sequencing data at 1 or multiple time points. Saguenay-Lac-Saint-Jean is a multi-generational study. Is a study of multi-generational French-Canadian families, and we have extracted methylation data from 3 generations. CAMPAC is a registry of individuals with peanut allergy. My thesis has 3 main aims. First, I aim to assess the accuracy and applicability of the Huebert algorithm to methylation sequencing data and its utility as a quality control metric. Second, I will evaluate the accuracy of this algorithm in pediatric samples. And finally, I aim to identify age-informative CpG sites in my dataset using linear or mixed effect modeling, and then using those CpG sites to develop novel, more accurate epigenetic clocks using more advanced machine learning methods. Thank you.
Great, thank you. Our next speaker is Derek Tam. He's a member of the UBC Bioinformatics program under the supervision of Dr. Ali Carson and Dr. Andrew Roth. He will be speaking to us today about predicting blood cell lineage by active DNA. Speaker, you may begin.
Okay, so hi everyone, I'm Derek, and today I'm going to be talking about tracking clones in blood cancer. So it's well known that your blood works incredibly hard to protect you from infection and to transport oxygen throughout your body. The average person will make over 200 billion red blood cells and 10 billion white blood cells every day. All of these blood cells in your circulation originate from a small pool of stem cells in your bones, and I've represented them in the timeline at the top of the slide. So as these stem cells grow older and we move right along this timeline, they'll accumulate mutations that occur during division, like the red and green cells. If the wrong mutation occurs in the wrong place by chance, a specific stem cell may acquire the ability to create blood cells that survive longer or divide faster. When this happens, your blood will disproportionately become composed of cells that originate from this mutated stem cell. While this isn't necessarily a problem, if a second mutation occurs that causes the stem cell to produce altogether dysfunctional blood cells, which are represented with the blue outlines, your immune system can become compromised. And in a nutshell, this is leukemia. So after someone's diagnosed with leukemia, they'll often undergo treatment that attempts to eliminate the problematic stem cells and the dysfunctional blood cells that they created. So while this is mostly effective, some cells, like the green ones, will escape treatment. The patient's condition will improve for some time, but soon the stem cells that survived will begin to divide again and take over the blood, with the added caveat that they're now resistant to that treatment. I'm very interested in this specific situation where leukemia relapses, specifically in determining what, if any, differences exist between these stem cells that survive and those that succumb to treatment. The problem is that it's impossible to figure out how one blood cell in a sample is related to another, as they're all identical. Or are they? So one neat trick that your blood cells don't want you to know is that they actually harbor little genetic breadcrumbs within their mitochondria, or your cell's powerhouses, which contain their own DNA that they're responsible for copying themselves. Unfortunately, or fortunately for us, they're not as meticulous as our own DNA copying machinery, which can result in up to 10 times as many errors. When these cells divide, they'll pass on those errors, generate some more of their own, and then pass those on too. So what this means is if we're able to see what these mitochondrial mutations look like in each individual cell, they contain a complete history of where the cell came from, as well as to which other cells they're related. While this problem might now seem quite simple, hopefully because of my excellent hand-drawn cartoon, the mitochondrial DNA is still over 16,000 characters long, and all these mutations need to be untangled computationally to rebuild what is essentially a massive family tree. Using a variant of single-cell DNA sequencing, we can identify regions of DNA that are active and simultaneously sequence the cell's mitochondria. By comparing these samples from before and after treatment, we're trying to see if any of the cells responsible for relapse were already present and hiding before treatment. As the project currently stands, we've confirmed that this approach does work, and I'm now looking at patient samples where we can apply this technique in a real disease setting. Thank you for listening, and I'll be happy to take questions later.
Thank you very much. All right, our next speaker is Emily Trudeau, under the supervision of Dr. Mary Burby. She's a member of the UBC Department of Botany and will be speaking to us about evolutionary insights into ancient plant-fungus relationships from secreted enzyme profiles of early diverging fungi. Speaker, you may begin.
Yeah, thanks, Rhiannon. So I'm going to talk about how I plan to use bioinformatic tools to infer ancestral ecological relationships between plants and fungi. Despite being ubiquitous and ecologically important, fungi are some of the most overlooked and understudied organisms. Fungi provide a range of vital ecosystem services like nutrient cycling and soil composition, and without them, the world would look a lot different than it does today. Different fungi can have different nutritional lifestyles. Some are plant pathogens, others are decomposers, and some form mutually beneficial mycorrhizal symbioses with plants. These nutritional lifestyles in fungi provide the underlying context for the ways in which plants and fungi associate with one another. To interact with plants, fungi secrete digestive enzymes that break down components of the plant cell wall. And these enzyme profiles correspond to the plant cell wall components that are capable of being degraded as nutritional substrates by fungi. So we know that the plant-fungus association is ancient, dating back to even before plants emerged onto land, shown in the middle panel by this timeline of early evolution that depicts ancestral green algae and fungi interacting in freshwater environments. And there's fossil evidence that even the earliest land plants formed mycorrhizal symbioses with fungi. But what we don't know is what kinds of digestive enzymes ancient fungi may have had in their arsenal around the time plants were first making their way onto land, and what role fungi may have played in the development of the first terrestrial ecosystems. My research aims to relate the evolution of digestive enzymes in early diverging fungi to the evolution of plant cell wall composition. To accomplish this, I'll be using bioinformatics to track patterns of molecular evolution in enzyme encoding genes using genomes from early diverging fungi that have recently been sequenced. Sequences that code for these genes will be recognized by their protein domains, which are the functional units of proteins that tend to be highly conserved. Fungal genomes will be analyzed to identify genes that are homologous and have a common evolutionary origin. I'll then reconstruct phylogenetic trees for these gene families and relate them to the organismal phylogeny to infer patterns of gene expansion and diversification. My work will provide insights into the genomic basis for fungal nutritional lifestyles and improve our understanding of ecosystem services performed by both modern and ancient fungi at a time where learning to cooperate and work with nature is crucially important. Thank you.
Thank you. All right, on to our next speaker. We have Jeremy Fan, under the supervision of Dr. Stephen Jones and a member of the bioinformatics program at UBC. He will be talking about merging, annotation, visualization, and summary of structural variants with Nanopore data. Speaker, you may begin.
So my work focuses on a particular class of DNA modifications called structural variants. And so these represent large-scale changes within our genomes, typically upwards of 50 base pairs as a cutoff. They're known to have disproportionately large roles relative to their abundance in the biology of diseases. But how do we go about detecting these variants in the human genome? So traditionally, DNA would be broken down to small fragments, and then we try to piece together these fragments onto a reference, very much like putting together pieces of Lego together to build a ship, using the picture of a ship on the instruction manual as reference. A limitation of this technology is that often the number of bases within each fragment are small, typically in the hundreds. And as you might expect, capturing a structural variant that is 10,000 bases long with only 100 base segments might prove challenging. So I'm interested in a specific type of sequencing approach called nanopore sequencing, whereby nucleotides pass through a protein instead of being broken down into these small pieces. And unlike the traditional sequencing technique, this approach allows fragments that are tens of thousands of base pairs long, which give more granularity into structural variants. So my project is to integrate a pipeline called MAVIS, which stands for Merging, Annotation, Visualization, and Summary of Structural Variants for long reads data. And so MAVIS is a post-processing pipeline for structural variants. And the overall goal of this tool is to seamlessly generate a meaningful annotated report to physicians and collaborators about structural variants found in a sample or set of samples. So once a tissue has been sequenced and structural variants called by softwares, in our case, let's say software 1 and software 2, calls can then be merged together. For instance, perhaps software 1 found a deletion at chromosome 4 at position 10,000, and software 2 found the exact same thing. If they happen to be exact matches, MAVIS will remove one of them. We can then validate these structural variants by taking the end of those variants and reconstruct them using the evidence that was used to generate them originally, just to make sure we can recapitulate the ends of these variants to make sure that they are what we think they are. And finally, we have the options, users have the option of illustrating some of these structural variants, particularly those which can be complicated. We can also cross-reference structural variants with known variants in different databases to generate an annotated report for users. So wrapping this up with an example, physicians, when deciding drug treatments, can then refer to the MAVIS generator reports to check, for instance, if a certain structural variant might impart resistance to a particular treatment option. So and that's basically the pipeline for MAVIS. And thank you everyone for listening, and I'll welcome any questions later as well.
Thank you. Our next speaker is Artie Kushner, under the supervision of Dr. Can Duk at the UBC Department of Bioinformatics. He will be speaking to us today about ribosome.xyz, the database for ribosomal structures. Speaker, you may begin.
Hey folks, so this is less of a research project than I think it's more of an infrastructure project. Over the last year and a bit, I've developed a database for ribosomes, ribosomal structures to be precise. We aim to be a central point of access to organized data about ribosome, whatever you can think of. We want to be able to provide that. We are trying to address something that PGB being the protein data bank, the big old warehouse for structural data. I think they have they're not able to provide the data of the granularity that we could. So just some background, PGB has about 200,000 deposited models. These are different proteins, some strands, some RNA, some larger molecules. Of those 200,000, there are about 1,500 ribosomes. But by mass, only ribosomes and viruses are they're by far bigger and larger than every other molecule in there. much more complicated. What we do is we take ribosomal structures and we process them and provide better categorization for each subcomponent. So, for example, on the slide, you can see that ribosomes are constituted usually of polypeptides, which are your proteins, polynucleotides, which are RNA, and non-polymers, which are usually ligands or some other antibiotics or ion clusters. That's usually where PTB's categorization ends. What we do is implement some custom classifier for each type of molecule, for each type of let's say subcomponent of the ribosome, based on the MMC format, crystallographic format, and so in the end we have a repository where you can grab classes of these molecules as opposed to downloading structures from PTB manually and parsing them and trying to muck about with the naming and different authors annotations which are inconsistent. So this is a big standardization effort. I think that about wraps it up for me. But I guess I'll take my remaining time to just complain about what people usually complain about in bioinformatics, being the
time's up. Please stop.
Oh, good.
Thank you. Thank you for speaking, Artem. I think we're just going to take a 1-minute break before we proceed to the next speaker. I'm going to leave this lovely slide up for the moment where we just want to check something going on in the back. Thank you for your patience. Okay, got all that covered. Thank you so much for your patience. I'm going to move forward. Our next speaker is Hans Ghezzi, under the supervision of Dr. Carolina Trupini with the UBC Bioinformatics program. You'll be speaking to us today about understanding the impact of gut osmolality on bacterial abundance and growth. Speaker, you may begin.
Hi, thank you so much for the introduction. In my research project, I'm interested in learning more about all the microorganisms that live in our guts, which are collectively known as the microbiota. In the last decade, these microbes have been increasingly linked to both health and disease, including important conditions such as inflammatory bowel disease, colorectal cancer, and other metabolic disorders. These diseases are known to drastically impact the gut ecosystem, and they change parameters like pH, temperature, and osmolality. And as a result, they may create an environment that is inhabitable for certain gut microbes. In particular, in my project, I'm interested in the osmolality, which is how much of a substance is dissolved in the gut. Osmolality can be quickly changed by laxative use or food malabsorption, and as I said, by other diseases. And it has a really big impact on the gut microbiome. A paper by my supervisor, Dr. Trupini, showed that a simple laxative treatment was able to cause the extinction of an entire bacterial family as soon as 3 days of treatment. This is a major change, this is a massive change. And it may be hard to appreciate because, well, we can't really see the differences. But this is a big change. And one way that we can appreciate it is by thinking about climate change. You know, in the Earth, there can be an increase in temperature. And as a result, the environment may be inhabitable for certain living organisms. For example, an increase in temperature may cause the death of bears, and this is comparable, the extinction of an entire bacterial family is comparable to the extinction of all bears on Earth. This is a massive change, as you can see. And in my project, we're trying to understand whether we can predict species abundance based on their environment. Just to link it back to climate change again, being able to predict bacterial abundance and growth in the gut based on osmolality would be the equivalent of being able to pinpoint exactly, for example, how many parrots there would be on Earth based on the temperature of the environment. As you can imagine, this would be a really important aspect of new and upcoming microbial therapies, including probiotics or FMT, fecal matter transplant, because we would be able to design new therapies that can have clear and expected outcomes on microbial colonization. And as a result, we will be able to revert this environmental changes caused by disease. And ultimately, this would be one step ahead towards our ability to treat and even prevent diseases in humans. Thank you so much to everyone for being here tonight and everyone at Immunology Lab.
Thank you. Great. Our next speaker is Ishika Luthra, under the supervision of Dr. Carl Huebert. She's a member of the UBC School of Biomedical Engineering and will be talking to us about predicting how single nucleotide polymorphisms change gene expression using deep learning in induced pluripotent stem cells. Speaker, you may begin.
Gene expression patterns control many different systems within our bodies, but our knowledge of how genes are expressed and controlled in humans is far from complete. So the regulatory mechanism between genes works in very complicated ways, and we have proteins called transcription factors that bind to sequence-specific sites in our genome, and they regulate the expression of nearby genes. Studies have shown that the gene expression landscape is partly controlled by the DNA sequence. So disrupting a transcription factor binding site can affect the binding and therefore influence the expression of a gene. Each of us has a unique genome sequence that codes how our bodies function. And we have random single base pairs that are occurring within our body, but most of these minuscule changes luckily do not alter our health or development. Occasionally, one single base pair out of the 3.2 billion in the genome will become mutated and cause a change in a gene's expression. So some SNPs that cause changes in a gene's expression lead to negative consequences and can cause diseases like cancer, Alzheimer's, or diabetes. I'm extremely interested in studying these changes and figuring out how they can help us uncover what is causing these diseases in the first place. So due to the large number of SNPs, we need to use models to help us understand which SNPs are the causal ones. So to understand the relationship between DNA sequence and gene expression, I plan to use a machine learning model called a convolutional neural network. So convolutional neural networks have been shown to be extremely powerful in capturing the complex nature of the genome, and they're also really good at handling large amounts of data. But because this is such a complicated task, we will use a machine learning trick called transfer learning. So transfer learning takes a model that has already been trained to predict how different transcription factors are binding to locations in the genome. And we will use this to predict how SNPs are affecting binding. So because we know that this model works and has been validated experimentally, what we're going to do is add more layers to the model, keeping the first section of the model static. So the last few layers of the model will learn the relationship between binding and expression. Once we have a fully trained model, we can predict how gene expression is changing based on only input sequence alone. Models like these allow us to systematically predict the effects of any SNP without the need for expensive and time-consuming lab experiments. With enough computational power, we can theoretically link the expression changes caused by any base pair in any location of the genome. Further, we can annotate how they influence traits and which of these mutations are leading to disease. Further, we can apply models like these to study rare disease phenotypes where the cause is unknown and the SNP is potentially occurring in a very small population of people. In conclusion, understanding the genome at a base pair level gives us the power to create targeted, personalized medicines and treatments. Thank you.
Thank you. On to our next speaker, we have Christina Daly, under the supervision of Dr. Ryan Morin. and a member of the SFU Molecular Biology and Biochemistry program. She'll be speaking to us today about PureDNA, a novel tool for estimating tumor purity from liquid biopsies in non-Hodgkin lymphoma. Speaker, you may begin.
Thank you so much for the introduction. So most types of non-Hodgkin lymphoma develop from the transformation of mature B cells in the lymph node. Lymph nodes are present all over your body, and so this cancer's development isn't restricted to one location. The most common and aggressive types of non-Hodgkin lymphoma are phenotypically and genetically diverse, where patients with the same diagnosis might not actually respond to the same chemotherapies. So because of this, patients can need different courses of clinical management, so early diagnosis, treatment monitoring, and response assessment are really important to an improved survival outcome. Cancer cells release cell-free DNA into the bloodstream through a process called programmed cell death, known as apoptosis. Cell-free DNA is DNA that's not contained inside of a cell and just kind of floating inside of a patient's bloodstream. Circulating tumor DNA is a highly fragmented type of cell-free DNA that comes specifically from the tumor. So extracting cell-free DNA from the blood is known as a liquid biopsy, and this is a non-invasive method to identify genetic and epigenetic changes in the patient's tumor, especially compared to a traditional tissue biopsy. Liquid biopsies contain DNA from both tumor and healthy cells, and since mutations can be present in both of these cell types, it's really important to figure out which variants are directly from the tumor, since this can affect how clinical decisions are made. So this is called tumor purity, where the amount of cancer cells in a tumor sample are quantified. Estimating tumor purity is actually really difficult since many sequencing and biological factors can affect this estimation value. Many programs exist to estimate tumor purity, although the majority only use one data type. So, for example, targeted or untargeted methods like qPCR or whole genome sequencing, and not many are actually directed towards ctDNA. So based on this, there's a gap in the literature where no programs actually exist to quantify ctDNA levels using genetic variants that are derived from both these targeted and untargeted approaches. So to address this, I've developed a novel bioinformatic tool called PureCTDNA, which stands for the purity estimation of circulating tumor DNA, that implements both copy number variants and somatic single nucleotide variants from two different data types, low-pass whole genome sequencing and CAP-seq. And this program leverages variant allele frequency, ploidy, and copy number to estimate the level of ctDNA from somatic variants in a blood sample. And along with an estimate and summary table that's output for every sample that you enter into the program, I've incorporated different parameters to account for unreliable copy number calls, as well as optional plotting. When comparing concordant purity estimates from tissue biopsy genomes to pure ctDNA values, a high positive correlation is seen, with additional validation efforts that are kind of still going on for more extreme cases. Ultimately, pure ctDNA will help improve the non-invasive treatment monitoring and relapse assessment for non-Hodgkin lymphoma patients. Thank you so much for listening.
Thank you, and my apologies for butchering the name of your application.
That's okay.
All right, on to our next speaker. We have Luke Trinity under the supervision of Dr. Hasna Jabari. He's a member of the University of Victoria Department of Computer Science and will be speaking to us about COVID-19, coming back to the fold. Speaker, you may begin.
By now, we are all familiar with this picture on the middle left of the screen. COVID-19 is the disease caused by the SARS-CoV-2 virus, leading to the current pandemic. What you may not know about SARS-CoV-2 is that it is an RNA virus. RNA is similar to DNA, except that where DNA is double-stranded, RNA has only a single strand. That makes RNA a lot like a piece of string. And if you've ever put a piece of string in your pocket, you know that it is guaranteed to fold into a knot. So if SARS-CoV-2 is an RNA virus, and RNA is like a string that can fold into a knot, you may guess that if SARS-CoV-2 causing COVID-19 is folding into a knot, it could be very important. Inside of this spiky orb that is SARS-CoV-2, there is a string of RNA. The spikes trick your cell into letting the orb inside, where it opens up and releases the string of RNA into your cell. Once the viral RNA is in your cell, your body thinks this new RNA is just like any other normal RNA you have. We have many RNA in our cell that send messages and perform various functions. Unfortunately, this means that your body will unknowingly translate the SARS-CoV-2 RNA into proteins and help the SARS-CoV-2 create more spiky orbs to send out and infect other cells. But there may be an Achilles heel to this process that can stop the virus in its tracks. If we look at the SARS-CoV-2 RNA from start to finish, there are actually two main sections shown here in blue and orange in the bottom left. Many different scientists are intensely studying the small area where these two main sections meet. At this location, the RNA has folded and knotted itself in a particular way to facilitate a crucial transition between the major sections. There is substantial evidence that if we can disrupt the folding of the RNA at this location, even slightly, it could be a life-saving treatment for those afflicted by COVID-19. I am using computational methods to predict the folding of the RNA at the specific location. If you look here at the inset image to the right, this is a simplified version of the RNA folding taking place. In image A, you can see dotted lines linking the first and third sections, and also the second and fourth. In image B, here you can see what the RNA looks like after it has begun folding. The first and third sections are now paired together. We call this a stem, and the second section now forms a loop. In image C, you can see that the second section is now folded to meet the fourth section, completing the knot at this location. The most exciting part about this work is that many different viruses use knots like this to function. The focus of my research is on identifying the most likely initial and subsequent folds, and this applies to Omicron and all variants of COVID-19. We hope to not only contribute to stopping the current pandemic, but also any emerging virus in the future. Thank you so much.
Great, thank you. Our next speaker is Sarah Dada, under the supervision of Dr. Stephen Jones. She's a member of the UBC Bioinformatics program and will be talking to us about the use of Oxford Nanopore long-read genome sequencing for precision diagnosis and treatment of individuals with autism spectrum disorder. Speaker, you may begin.
Autism spectrum disorder is the most prevalent childhood developmental disorder, diagnosed in 1 in 66 children in Canada, with 3 of 4 of those being male. It is diagnosed by repetitive patterns of behavior and deficits in social communication. If you've ever hung out with a group of people with autism, you might have noticed that a person's autistic traits look a little different from each other. Some people might wring their hands, some might be completely nonverbal, and others may not laugh at your joke. Or maybe you're just not funny. But autism truly is a spectrum. Appropriately, each individual has a different cause for their autism as well, and that is likely due to differences in their DNA. All living things are made of cells, and inside each cell there is genetic DNA. DNA is a string of A's, T's, C's, and G's in various orders that give instructions for how the cell should function. And when this order is disrupted, it can cause disease. We can use computers to look closely at this DNA sequence. Large changes in this ACTG coding structure of the DNA are known to be associated with autism. But until this very point, these large DNA changes were difficult to discover. Therefore, we needed to use new technology to find out what DNA changes were associated with autism. This is my project. Previous technology used in research was called short-read sequencing, which gave a smaller picture of DNA changes. My project is to use now long-read DNA sequencing, which gives a larger and more accurate picture of large DNA changes. I'll use this in children with autism at BC Children's Hospital. This will solve larger and more complex DNA changes that may not have been implicated previously in autism. Short-read sequencing, which gives a small picture of DNA, leaves out some information. Here's an example. Imagine you have a piece of DNA that's supposed to look like the picture under short-read with the computer. But your analysis shows that someone with autism has DNA that looks like the picture under the human. It looks like a simple switch of 2 DNA bases, C and T. As a researcher, you're a little unsure of the impact and whether or not this change is involved in autism. But imagine later you get long-read data of this person, which gives a much bigger picture. You realize you've been tricked. What you thought was a small change was actually a very large change of an entire piece of DNA. It flipped, and this could have a much bigger impact. Long-read DNA sequencing can help us infer a lot. It can show large duplications. It can even show you if there are marks on the DNA to silence it and make it unusable for the cell. This information will let us uncover secrets of DNA that may lead to breakthroughs in how autism arises, better diagnosis and treatment, and why autism is different between people. It is crucial in our understanding of DNA and other diseases and how it makes us unique. Thank you.
Thank you. On to our next speaker. Taylor McCole, under the supervision of Dr. David Clark, is a member of the SFU Biomedical Physiology and Kinesiology Department. He'll be speaking to us today about kinetic modeling of the cellular signaling controlling skeletal muscle protein synthesis following protein ingestion. Speaker, you may begin.
Have you ever thought, how did I get out of bed this morning? Or how was I able to walk to and sit in my seat? Well, these day-to-day physical activities rely on your skeletal muscles, and more specifically, having a sufficient quantity of skeletal muscle mass to allow for adequate strength to perform these activities. Many factors contribute to the maintenance of skeletal muscle mass, of which the one factor that my research is particularly interested in is protein feeding. Protein feeding acts to stimulate cellular signaling pathways, with the ultimate outcome being increased muscle protein synthesis. Essentially, protein consumption tells your cells to build more muscle. Now, the mechanisms by which protein feeding governs muscle protein synthesis are well characterized at the individual level, but how these individual mechanisms interact to produce the observed muscle protein levels remains unknown. Furthermore, investigating how these individual mechanisms act at a systems level is made difficult because much of the muscle protein synthesis data comes from various data sources that are not easily comparable. However, mathematical models can be developed to solve this problem. Mathematical models are essential bioinformatic tools for integrating biological data from disparate sources and investigating the complex interplay between governing mechanisms at the system's biology level. Here, I developed and validated a mathematical model of the cellular signaling controlling skeletal muscle protein synthesis following protein synthesis in healthy adults. I developed this model by amalgamating modified versions of previously published models of the cellular signaling, hormonal response, and amino acid kinetics following protein ingestion. My model synthesizes knowledge from disparate data sources into a coherent framework and successfully reproduces the magnitude and kinetics of muscle protein synthesis. Our model represents a first-generation quantitative hypothesis of the dynamics of protein translational control in healthy adults, that being adults who are able to maintain their muscle mass throughout a 24-hour period, provided they receive adequate feeding. However, muscle mass is not always maintained over the course of a day if there are dysregulations in the cellular mechanisms controlling muscle protein synthesis. Over time, this dysregulation can lead to a loss of skeletal muscle. Dysregulations in the cellular mechanisms is commonly observed in older adults as part of the aging process, which can lead to a loss of muscle mass, overall strength, and reduced quality of life. Future analysis with our model promises to identify the cellular signaling components responsible for this age-induced dysregulation and discover potential sites for therapeutic interventions. If successful, populations both young and old will have the muscle mass required to perform day-to-day physical activities like getting out of bed or walking about that most of us take for granted. Thank you for listening.
Thank you, all of you. A big round of applause to all of our speakers. Those were fantastic. I really enjoyed myself, and I feel like I learned a lot. Speaking of which, now it's a chance for kind of the audience to kind of put in their vote for who they really appreciated. I'm sure we were all appreciated, but who was your favorite pick? You can go to the link on the page. One of the moderators will put it in the chat as well. We'll take a few minutes now to give the audience a chance to vote and speakers a chance to decompress, let alone let the judges actually, they can take a moment to enter all their tallies into our processing sheet. Okay, well, we can still let some of those trickle in, but while that's happening, how about we get going with some questions and answers? So audience or judges, in order to ask questions, you can either raise your hand and we'll call on you to voice a question, or you can type it in the chat and a moderator will voice it for you. I'm not seeing anything so far, so maybe I'll take a moment. And I guess I can even jump around the slides if we want to. Let's start with David Huebert. Are you available to take a question?
Yeah.
All right. How would you validate the interactions proposed between A through G and BIF?
So we did multiple ways of validating it. The first way was that seeing as we were looking at so many species, we did validation methods by getting apobecs that don't have any interactions with VIF to interact, like see if the interactions would still hold or not. And we went back and forth with checking what will happen if two proteins that we know don't interact properly interact with one another, ones that we know can but don't do it well. So we did a lot of crossing back and forth to figure out how different interactions can happen. And on top of that, you might have seen it in the actual slide. There's a heat map there, and we were able to actually figure out percentage-wise how often every single amino acid within these structures could be interacted with. And some of this goes back to validation that we were able to create through multiple previous papers as well.
Hey, thank you for that.
Okay, I'll ask the next question. Is Artie Kushner here? Artem, are you still here? Okay, he's not responding, so I'll move on to the next speaker. Armita. Armita, are you there?
Hi, yeah.
So my question for you is, how would RinsLR determine that the inserted sequence is actually normal? Like, for example, when you determine that there's an insertion happen at a particular region in the genome, how do you determine whether it's novel or not?
Yeah, that's a good question. So from what we have done so far, we take the repeats, like simple repeats that are already detected in the genome through different tools or databases that are already available publicly. And we can check the insertions that we have found versus them to rule out the simple repeats that would not be a novel sequence insertion.
That's great. I guess I'll continue. Oh wait, there's a question from the chat.
I think, yeah, Charlotte has her hand up as well.
Yeah.
Oh, thanks. I have a question for Asfar. You mentioned in your talk about measuring the transcription factor binding, how it's affected with these sort of variations. And I wondered what tools you used. Did you sort of model it structurally and use AlphaFold, or did you use quantitative tools?
So I haven't done it yet, but the idea is to build a mathematical model that will capture how known transcription factors in the targeted region is getting affected due to the sequence change in that area.
Awesome, that sounds really cool. Thank you.
Okay, I don't see anybody have their hand up, so I'll ask the next question. Artem, can you please turn on your mic? Okay, he's not responding again, so I'll move on to the next speaker. Jeremy, are you there?
Yep.
So in this talk, you basically talk about Mathis and how it works with nanopore sequencing data. Would it work with PacBio data at all?
Yeah, so it would just, I'm guessing for PacBio you use different structural variant callers, and so you can just I think currently right now there's even like a general converter for different callers, so I think it would work. And if it doesn't, I can always like write in like a script or something in a module to convert the output from those different callers.
That's great, thank you. Do the judges have any questions for the speakers?
I can ask one question. I think this is for Derek. I'm just wondering, while you presented the work for cancer, can the same approach be used to study microbial populations?
That's a good question. I believe it could be, as long as it's actually been a while. I don't remember if microbes have mitochondria, but if they do, then technically the method should work. The idea is that if you use single cell ATAC-seq or an assay for transposase accessibility, you can sequence mitochondrial DNA because they don't have like chromatin organizing structures. So that's why it usually comes up as a contaminant if you're not interested in mitochondrial DNA. But for these purposes, it's actually great. So yeah, in theory, this could work.
So this is a question, and I'm sorry if I missed a response to a question earlier. I was busy typing in scores to the right spreadsheets. But this is a question for Luke, if Luke is still available.
Yes, go ahead, go ahead.
So Luke, is there, there's lots of different RNA structure callers out there, and so how are you picking which ones to use?
That's a great question. So what we're doing is we're building off of what's called the hierarchical folding hypothesis. So as you say, there's a lot of different ways we can look at the structure. And for example, like we're probably most interested in the final structure, or the three-dimensional structure. And so we're hoping to contribute to that by increasing the accuracy of how we understand the first folds, right? So having a more full understanding of the complete process to the final structure. So for example, if there's a few different most likely initial folds and we can understand that better, if we're trying to disrupt this knot from forming, we really need to have a complete understanding of the structural landscape. And a couple of the other things that I'll quickly mention is related to incorporating external data sets. So of course, there's a lot of probing on the COVID, viral genome. And so how are we incorporating that? And so that's part of our process is, there's a lot of different data sets out there and including those in our predictions.
Great, thank you. Do you have any follow-ups for that, Dr. Wasserman? All right, I guess we'll move on to
Just so that everyone knows that we have a little debate going in the background between Will and I, because we have everybody, there's so many people who did so well that we're squabbling over our scores.
All right, well, great. We'll give you some more time and keep asking some questions. Actually, Ishika, if you're available, I have a question for you.
Sure, go ahead.
So it's kind of a double question. Does the model only work with gene sequences? And if so, what about SNPs in the intergenic regions?
Yeah, so theoretically, we could input any DNA sequence, but SNPs that occur in the non-coding regions or like are acting in trans that are really distal, those are usually pretty difficult to predict, mostly because I think when you train the model, it kind of blows out of proportion if you have really, really long input DNA sequences, but that I think that is more of a computational limitation. There's some new approaches, if you're interested, it's called the Informer paper, and it's able to do, I think, 200 kilobases long input DNA sequences to predict expression, which is really cool. Yeah, but that's a different kind of machine learning model.
Great, thank you.
Mm-hmm.
I guess, how about Sarah? Are you available for a question?
Hello.
Hi. Here's with your autism spectrum disorder study, would like the parents also be sequenced at any point to be part of the analysis?
Yeah, it's actually a pretty interesting thing because we're making a larger cohort now, and it would be nicer to be able to just sequence the children. But then if we're interested in any imprinting at all, we would never be able to find out whether the father and mother's methylation patterns are implicated without the parents there. So I think right now it's one-offs, but when we get more, we're probably going to have to incorporate the parents as well, so we know what their SNPs are. Who's it coming from?
Great, thank you.
Thanks for asking.
Right, I think actually, Emily Trudeau, are you available on the call?
Yeah, I am.
Great. So what about like fungal enzymes that degrade antifungal compounds in plants? Like, is that relevant to your work?
You mean you're asking about fungal enzymes that degrade antifungal compounds?
Yeah.
Okay, that's interesting. No, I'm mostly just looking at enzymes that degrade components of the plant cell wall. So those would be involved in like fungal nutrition. So in order to get into the cell, they need to first break down the cell wall, which includes like pectin and cellulose and hemicellulose. So those are the main components that I'll be looking at, not really any enzymes related to like antifungal components.
Thank you. Sorry, it wasn't a more complicated question.
Christina Daly, are you there?
Yeah.
So in your talk, you mentioned that you mainly focus on SNPs, am I correct?
Single nucleotide variants, yeah.
So how about indels and XVs? Like, wouldn't those be important as well?
Yes, so basically how my program works is that there's two inputs. So we have the copy number variants, but also the single nucleotide variants. And it's just specifically from variant calling programs, which output math files. And so indels are included in those as well. I just single nucleotide variants, but yeah, anything that's called by a variant caller, such as like I use Sage, for example, but I know a lot of people use Strelka and Mutect and things.
Okay, thank you.
Does anyone have any other questions? Anyone from the audience, something percolating?
Do the judges need more time?
Oh, I think the judges are all ready, actually. So I can, yeah, I can, if you guys would like to do the honors, I'm just going to put on the slides, but I can also announce them, whatever your preference is. But we'll start with third and then second and then first.
Okay. I think it would be great if you announced it.
All right, I will go forward with that. Okay, thank you so much for putting in that energy and getting through the kind of final debate of it. So our third place winner is Christina Daly. Thank you so much. That's great. We will be following up with you. And then second place, Luke Trinity. Congratulations. And for our first place winner, we have Ishika Luthra. Thank you so much. Great job, all of you. Like everyone did a phenomenal job, but like absolutely wonderful. We will follow up with all of you after this to work out getting this, but thank you all for doing such a great job. On to our viewer's choice. We also have Ishika takes that away as well. Clearly, you can be very proud of what you brought in here today. We're really, really happy to have had you. Thank you so much.
Thank you, guys.
Great job, everyone.
Ah.
In the top three, the difference is by 0.5 mark, so it's really close call. That's why it took us a while to sort that out.
And then I guess I'll quickly announce the audience prize draw. We have two winners. We have Abdul Muntakim Rafi, my apologies if I pronounced that incorrectly, and Charlotte Barkley. We will also follow up with you with the email that you used to enter in the form. Thank you so much for participating and putting and engaging as an audience. We really appreciate it. Does anyone else have anything last things to say? Like panelists, you're more than welcome to take a moment. Yeah. All right. It sounds like we're all good. And yeah, we've got some chat going on, but I'm going to stop the recording and just once again, thank you to our judging panel. Thank you to our speakers and thank you to our audience. You really made this event happen and turn into a wonderful occasion. Thank you.